So I'm part of the Centre for Multiscale Theory and Simulation, CMTS, as well as the Computation Institute, and I'm largely based in Argonne. So it's one of these things where the attributions kind of vary from time to time. And for all I know at the moment, I'm technically on secondment to the CIA as well, so nobody tells me anything. So the first thing is, what is multiscale theory and simulation? The CMTS is really interested in coming up with techniques to rigorously bridge these length and time scales between different systems, such that we preserve the essential physics. So the multiscale problems typically have several scales of length and time. So I deal with molecular-based systems, and the interactions between the molecules will often influence what the sort of mesoscopic or macroscopic behavior of the system is. So a little example here, we have an actin fiber. Now actin's a really common protein. It's in all of your cells and it's super important. And fundamentally, it's a molecule. It's made out of a bunch of atoms that are connected together in particular ways. They have certain interactions, and it can be very expensive computationally because the actin filament itself is formed of lots of these subunits of actin molecules, and it's very big in a sort of cell scale sense. So we try to create simplified models of these systems so that we can really start to look at cell scale processes while still capturing the essential physics of the molecular level systems. So cellular biology is one of the major things that we're dealing with, because there's so many things going on inside your cell that we have a certain amount of information from in terms of experimental results, but we don't really understand the fundamental processes of work in some cases. So again, here we have the actin, and we have several thousand atoms that are present in this molecule, and it comes together to form a sort of filament, long and thin. We can't really simulate these things very well computationally because they're very big. There's lots of atoms involved, and the sort of time and length scales that we need to access with these simulations aren't really possible with the conventional detailed simulations. So we'll generate simplified models of these things which capture the essential physics. And by using the simplified models, we can really start to push up the simulations to very large scales, the sort of scales that will give you some sort of an indication as to what's going on in the cellular level. This idea of sort of emergent behavior of these small level interactions giving you an overall behavior. And one of the things that we're really interested in is biology, because it's such a nice target system for these things. I've talked about the actin before previously. The actin itself isn't just these long, thin fibers. It's actually branched like a tree. There's lots of complicated things going on. And these are all processes you can't really access with the conventional detailed simulations, but we're trying to create these sort of simplified models and let us study these in more detail. Lots of actin systems. And my particular little sphere of influence, if you can call it that, is retroviruses. In this case, HIV.
Do you mind putting on a lot of yours so that we can hear you?
Listen. Is that working a little bit? Is that working?
Yeah, it's working. Thank you.
Okay, right. So if you think about how you actually represent these systems, there's a sort of hierarchy of work. And at the bottom here, the most detailed sort of model you have is you have all of the atoms present, and you have all of the water present, and it's pretty much a full detailed system. So you don't have quantum information in there. It's still a classical system, but there's lots and lots of information in there. To make the simulations a little bit more efficient, one of the things that you can do is remove the actual water. There's lots and lots of water in biology. If you can remove those explicit water molecules, there's just less degrees of freedom. Everything becomes faster, it's a more efficient simulation. Moving up a little bit more, we can remove some of the degrees of freedom, like the explicit hydrogen atoms in there and all these things, to generate an even more simplified model, which again lets us access these larger timescales, larger length scales. But up at the top, we have this idea of ultra-coarse-grained models. So this is one of the things that really motivated the work we're going to talk about today. Because they're dynamic, when we try and get rid of all of this information that we have in the detailed model, we need to somehow capture the behavior of those molecules all over again with fewer degrees of freedom. And if everything's fixed in the simulation, so we have a simplified representation of this molecule with a certain sort of interaction assigned to each of these sort of quasi-atoms in there, there's lots of things that we can't properly capture. So there might be environmental properties going on where the actual interactions between these molecules will change depending on what the local environment is. And if everything's fixed, you can't capture this sort of behavior. So the idea of these sort of ultra-coarse-grained models is that the actual model itself can change its properties depending on what's necessary in the local environment. So we can have things like isomerization, protonation of all these molecules. We can have various substructures changing inside of these beads. Lots of fun things going on. So it allows us to use these simplified models while still capturing more of the actual information that we need. Which all sounds great in principle, but when you actually come around to trying to apply these sorts of models, you have real problems with the conventional software. So Molecular dynamics, where you're actually integrating Newton's equations of motion for these molecular systems, has been around for quite a long time, but it's been around under a certain guise where you make some assumptions about the system that are no longer valid for these ultra-coarse-grained models. And the removal of the water, for example, means that you can quite often have these large empty regions inside the simulation. So you need to be able to balance the workload that you're doing evenly across all of these CPUs in a supercomputer, while still taking into account the fact that lots of the volumes inside this sort of overall simulation might actually be empty, so there's not really any work associated with them. If you're pushing up to these really, really big length scales, and Katrin's probably laughing at this point when I say really big length scales, because you know we have somebody who's an astrophysicist here, and I'm saying cells are big. But nonetheless, you need to be very careful about what you're doing with memory at this point. And also, when you have this sort of highly mutable simulation context, this idea that the ultra-coarse-grained models themselves can change their properties dynamically during the simulation, you need to be careful about how you represent the system and what assumptions you make in the actual modeling software, such that you can really deal with these dynamic systems. And we basically needed new tools for this. The sort of theory that's coming out of the CMTS is not a nice match for the existing software. And necessity is the mother of invention, so we generated an entirely new way of doing this. We're calling it UCGMD in a quite spectacular sort of failure imagination at this point. I'm not going to go into a huge amount of detail about the sort of algorithms that I'm describing here. We recently published a paper on them, so if there's any technical aspects that maybe you're interested in to do with how we deal with the network communications and various other parts that aren't really all that interesting in terms of the application but are important for the actual programming side of things, then this paper will be placed in all of things. Okay, so the first problem that I'm going to address today is one of load balancing. So we have these enormous supercomputers. We have the Mirror Illusion at Argonne, which has over 750,000 CPU cores. The Blue Waters machine at the NCSA is likewise very, very large. But the problem is actually writing the parallel software that will balance the work that you need to do evenly across these very big machines is surprisingly difficult. Particularly with things like molecular dynamics, because it's tightly coupled. You need to make sure that each of the CPUs has finished its own little piece of work at each time step before you can proceed. So if any one of these CPUs has too much work to do, if you haven't balanced the load evenly across everything, then you're limited by the speed of the slowest CPU, which is the one that has the most work to do. So if one of these CPUs is massively overloaded, your entire simulation will just slow down in a very systemic way. So with the conventional sorts of simulations where you have explicit water in there, you have a bunch of molecules of water in there too, you typically have a sort of roughly uniform distribution of particles in the simulation. And at this point, you can simply split your simulation up into roughly equal volumes and assign one of these volumes to each of the CPUs, and each of those CPUs will handle whatever work is needed inside that little subvolume. Provided you have the even distribution of particles, it's relatively straightforward to do, and you get the load balancing emerging kind of naturally from this sort of a system. So each of the CPUs has a roughly equivalent amount of work, and everything works quite nicely. When you use the UCG models and when you remove the explicit water in the system, what you find is you'll have little patches inside the simulated volume that have high particle density, and other regions that have low or even zero particle density. And at this point, chopping up your simulation into these roughly equal-sized volumes doesn't work anymore. You find that one of the simulation domains, for example, this one in the top right-hand corner, has way more particles than, for example, this one or even this one. So this will be the bottleneck. This is the point at which you all slow down while that CPU is waiting to complete its work. And the way that we got around this is actually a trick that we borrowed from astrophysics. We use the Hilbert space-filling curve, which is a sort of mathematical construct that will fill a three-dimensional volume with a one-dimensional curve. So it sort of weaves its way through this three-dimensional volume, filling up space as you go. And the advantage of using this sort of a system is that you can basically map the positions of the particles into the sort of lattice coordinates of this underlying three-dimensional curve. And by doing that, you can then partition up the curve into sections that contain roughly equal numbers of particles. So this is how you achieve the actual load balancing. A little illustration of it here is taking this slightly difficult unbalanced system. We weave our little curve through, and then we split up the curve into sections with equal numbers of particles in them. We also get a certain amount of compression in the load balancing, because typically you have more than one particle in each of these Hilbert cells. So when you send it to the load balancer and it figures out what performs which work, you're not sending the full amount of data. It's actually a very compressed form of the overall system. So this is quite nice in terms of the network communications as well. So to try this out, we took the worst system we could think of, and I've got a little schematic at the bottom here, and some sort of a biological justification for it. It's a flat plane of biolanes, sort of similar to a cell membrane, with two spherical biolanes that are positioned above it. And this is relevant for things like neurotransmitter release by synapses and things like viral infection, where you have these viral particles that actually merge with the cell membrane and release their contents into the cell. And we found that using this sort of load balancing approach, the space-length curves, even for really unpleasant systems like this, where it's just all empty space apart from the particles being really closely localized in certain places, we get really nice load balancing properties over 3 logarithmic decades of CPU core count. So we can go from 128 CPUs up to 250,000. And we get basically linear scaling by using this sort of a load balancing approach, even for really nasty systems like this. This is the sort of system that a conventional ND code would really struggle with. You would not get the nice scaling behavior for these things. Another aspect of this is the memory use. When you have these particular systems, they're interacting, the particles, if they're within a certain distance from one another, are actually exerting a force on one another. And the trick is knowing what's exerting a force on what in the system. The obvious way to do this is for every particle you have in the simulation, you look over all of the other particles that you have, you calculate what the separation between the particles is and whether or not they're interacting and what the force would be. That sort of an algorithm is of order n squared, which is a really bad idea when you have large numbers of particles. It just will not scale well at all. The way you typically get around this is something called a Verlet list. So each particle actually stores a list of the other particles that are sufficiently close to it at one MD time step to be interacting. And because you have this sort of spatial locality or temporal locality in the particle positions, they don't really move a huge amount in each time step. That list that you generate should in principle be good for several ND time steps, so you can sort of minimize the cost of building these lists over several time steps. But it's still fundamentally order n squared. So a further efficiency that you can, efficiency improvement you can make is this idea of link cells. So these are like little notional cells inside the simulation of roughly cell dimension, the sort of non-bonded cutoff between the particles. By mapping the particles into these little cells, you can quickly generate a Verlet list by simply looking at all the cells that surround any particular cell. And this speeds things up massively. Instead of having to cope your entire simulated volume with these little cells. And for conventional sorts of simulations, where maybe you're looking at tens of nanometers, hundreds of nanometers for these molecular systems, that's not a problem because you still have a relatively small number of these little link cells, so the memory overhead isn't particularly significant. When you're pushing up to really big simulations, like here I've got a mouse fibroblast with the little bar at the top showing you 20 microns, this becomes a serious problem. The conventional algorithms that just pattern the entire simulation domain with these link cells end up using huge amounts of memory. So the red curve here is showing you the sort of memory requirements for just a single particle simulation in the conventional ND code as we make the simulated domain bigger. So even though we've only got one particle in there, we quickly see the memory requirements skyrocketing just because of this link cell algorithm that's used to speed things up. And it's an example of why you need to take a step back sometimes and say the conventional modeling software does things in a certain way. But that doesn't mean that's the way that you should be doing it when you start pushing it to really large scales. By comparison, the ECG code that we wrote uses a sort of sparse on-demand spatial data structure. So rather than covering everything with these link cells, we basically only generate where they're needed. So we basically, fundamentally, we have a sort of dictionary or a map or a hash table, however you want to think of it, that's indexed using an integer triplet, which is the XYZ coordinates of these little lattice cells. And if there's nothing in one of those lattice cells, that data does not exist. So we managed to sort of really put a limit on the amount of memory that we're using. So for the same system here, the green line you can just about see here is the ECG code. Well, when we make the actual simulated domain bigger, it just uses the same memory. It's really decoupled from the amount of volume that we're simulating. And this is nice because it means we can cap the amount of memory that we need for these really big simulations. The final thing, and this is something that's been overlooked a little bit, the NSF and the NCSA are really quite excited about what we're doing with these modeling codes. And they're really focusing on things like the memory use and the scaling properties of the code. But that, to me, is not the interesting part of this. The really interesting bit about this code is the way we've written it is that it's very dynamic. It regenerates all of the internal data as it needs it. And we can motivate that sort of an approach with a system like this. This is actually the HIV viral particle. So when this is released by an infected cell, it's initially in this sort of conformation. So grey is like a little cell membrane that surrounds this viral particle. We have these sort of spoke-like molecules moving out from the membrane into the interior. And red here is the viral DNA, or viral RNA, I should say. And there's a sophisticated sort of set of processes that happens to take us from the image on the left-hand side to the image on the right-hand side, which is the mature and infectious viral particle. There's lots of things like enzymatic cleavage occurring there, and assembly of nanostructures that happens inside this viral particle to make it infectious. This is really the sort of stuff that the conventional codes do not like. The conventional modeling codes assume that whatever you put in at the start of the simulation, is basically what you have throughout the entire simulation. Your positions may move and the velocities may change, but fundamentally those particles, those molecules, remain unchanged. And that's not something that's appropriate for a system like this, that you have these significant mechanical changes occurring. So I've stressed the dynamics and molecular dynamics. We've eliminated this idea that we have anything fixed in the simulation. Everything's regenerated as and when it's needed. And this is built on the basis of this idea of template molecules. Typically, when you have these simulations, you have lots of copies of what's effectively the same molecule. Rather than repeat all of that information over and over and over again, we just define an example of that molecule. And the code will actually generate all of the internal information regarding things like bonds between particles, angle forces to hold structures where they should be, and all these sorts of things dynamically. And that's deceptively interesting, because by taking this sort of dynamic approach, we can basically put in lots of interesting chemical properties into the simulation that you can't do with conventional code. So things like multi-state models. The molecule that you place into the simulation doesn't necessarily have the same properties throughout the simulation. It can change dynamically. We can change the resolution of the molecules. If two molecules are close together, maybe you want it to be high resolution to capture the fine details of an interaction. But as they move apart, you want it to be slightly less detailed, so it's computationally cheaper. We can add and remove molecules from the simulation dynamically. This isn't a problem. We can do things like anabolic and catabolic reactions, where things are actually polymerizing or depolymerizing, or we have enzymatic cleavage and all these sorts of things. And this emerges naturally from the idea of using this sort of dynamic generation of the simulation data as we go along. And this gives us a distinct advantage for many of these sort of biological processes versus the conventional codes, because we can handle these quite significant chemical changes that must occur in things like the viral maturation process to really model these things on a molecular basis in such a way that we can try and get some insight into the experimental conditions. Okay, so wrapping it up, basically, the conclusions in the future work here is we've taken a step back and looked at what the conventional modeling software did for these chemical systems. We thought about what it was that we needed to do with the new theory that's coming out of the CMTS, and we realized that the existing code isn't really good enough, so it gave us an excuse to basically start from scratch and try some different algorithms that could solve many of the problems that already exist in these sorts of pieces of software. Future improvements, you need a slide like this. I'm trying to get some more GPU acceleration. So this is a great target for things like OpenCL Computation and things like this, because we could really concentrate the calculations we need to do very nicely onto single CPUs, single nodes. By doing this sort of thing, we can use GPUs really nicely. We can cram a lot of work onto a single node, so we're not having to move data backwards and forwards, and it will work really nicely, I hope. We did some quite clever tricks with some of the hardware on the network for the systems we've been testing it on, and it worked really well. Most people are familiar with things like MPI, and MPI works well up to a point, and then you start to see you have problems in terms of the network communications. That can really form a bottleneck in terms of how fast you can run things. We sort of sidestepped this by using low-level things like EMAP, AMI, and blue machines, and we got some great results in terms of really speeding up the communications. We want to try and put more of these ideas into the fundamental communications to make it even more scalable. Per particle load balancing. The thing is, with these ultra-coarse-grained models, the computational cost of doing the calculations involved in any particular particle is not necessarily constant. You may have a very simple interaction in one sort of particle, or something quite complicated in a different sort of particle, and the actual amount of CPU time required to do the work involved in those two particles might be quite different. So rather than just saying what's the particle density and the load balance on the basis of how many particles we have in various places, we can start to measure at runtime what the computational cost of the work is involved in each of these particles and molecules, and we can feed that into the load balancer instead, trying to eliminate this problem where you have really different fundamental interactions between the particles, we can still get good load balancing. And at that point, I think I'm running out of time, so shall we just hand it over, or are there any questions?
Yeah, questions. So when you run a simulation like the one you said, the HRDB one, so how do you know whether your answers are correct? What's the
experimental data. I'm a big believer in experimental data. I've seen way too much work from people who do computational simulations who say, and it looks like it does this. No, if that's not what you're seeing in the experimental data, then it's wrong. Okay, nature is not wrong. You can't just sit there and say, well, the experimental system do this, but my simulation says something else entirely. I think at that point it's a pretty safe bet that you're in the wrong. You've got to match it to experimental data. And to those ends, we work extensively with a lot of experimental people, X-ray crystallographers, electron tomographers, people like this, to really try and get the fundamental information we need. But in those cases, it's just a snapshot. What we're trying to do is fill in the blanks. We have the dynamic behavior. They may give you an image that says, this is what the final structure looks like in self-assemblies. So one of the ways you can do this is try your model from complete disorder, see whether or not it self-assembles into something that's experimentally correct. And at that point, you've got a reasonably good idea that your model's doing what it should be doing. But always go back to the experimental data, otherwise, I don't really know what it is that you have there. It's probably interesting in some senses, but it's not necessarily relevant to the physical system you're trying to model.
Can you get 4D experimental data, with time resolved?
Depends on what sort of timescale you're looking for. One of the problems with the coarse-grained simulations is when you remove detail from the model, the timescales change a little bit. You effectively get sort of enhanced timescales. So by looking at the raw sort of explicit timescale you're using in the simulation, that doesn't necessarily map to a physically realistic timescale anymore. So at that point, typically what you're doing is you're looking for experimental properties that have well-resolved sort of temporal characteristics, and then you measure them in the simulation too, and you attempt to map between the two sort of timescales that you have. That's actually an open problem. It's very difficult to do that sort of thing.
Okay.
Hello.
I have a question about when you do the parallel programming.
Sure.
You divided the whole system into different into small sections, right? So, like, how do you deal with the particles which close to the boundary of those sections? So, like.
That is a brilliant question. That's something that was actually quite tricky to handle.
Yeah.
When you have these sort of regular cuboidal domains that you put your particles into, it's quite easy to figure out what's going on across these sort of interfaces between CPU codes. If you have a particle that's right next to this domain that divides one of these little sections on one CPU, and one that's on another CPU, but they're next to one another across that boundary, you need to be able to calculate that interaction across the boundary. For these regular shapes, where you have these sort of cuboids, it's pretty straightforward to do. When you have the Hilbert curve, if you imagine that a section of that Hilbert curve is weaving its way through the system, if you just pick two arbitrary points here, a start point and an end point, you no longer have a regular volume. It has all sort of irregular edges and the boundaries all over the place. So figuring out what needs to talk to what in the simulation, how you're moving the particles around, becomes really quite difficult. And this is one of the reasons we're using the network-specific programming stuff, because we actually needed to somehow share this information in such a way across the simulation that everybody knew where everybody else was, even when you're dynamically load balancing and what you've been assigned in terms of the Hilbert curve is changing. And it's a little bit technical. It is covered in the paper that I listed a little bit earlier on, but that was something that was not straightforward to do. But we've got it working, and we've got it working quickly now by using a few of these tricks to do with the actual network hardware. It's not as straightforward as you might expect. It took me a little while to figure out how to do this and do it quickly. So it's a good question.
So, like in the kind of the probably assembling, like you calculate the interactions between the particles in different sections.
Yes. So yeah, basically you have this mapping in the Hilbert curve. You say CPU number 1 has curve indices of 0 to something. So when you detect that the particle is moving potentially close to one of these irregular boundaries you have, it's relatively straightforward to just look at the surrounding cells and map those into which other CPU actually owns that domain. Because remember, you have this set of slices in the Hilbert curve, so you can say, okay, the curve index this is this cell that's just a little bit outside the curve. I know who that is because I have this mapping from the sort of CPU ID into the little region of space. But it's tricky to get that running quickly. It did take us a little while to figure that one out.
John, there's going to be a question coming through our chat here in a moment.
Oh, okay.
So we're waiting for that. There we go.
How do we know that the transients of dynamics generated by the model are correct? Again, it's a good question. I'm basically looking at, to a greater or lesser extent, these sorts of equilibrium properties of many of these things. Now, at that point, the dynamics aren't quite as important. It's more so thermal quantities, so we're looking at the average properties. But again, it's a good question, and it's something that is an active field of research, actually, to figure out how you deal with the dynamic properties of these things. There's certain things that you can do, things like dynamic force matching to generate the actual interactions in these coarse-grained models, but it's still quite an early field here. We don't really have definitive answers as to how we ensure that the transient dynamics are correct. So I typically look at what the results are in terms of the structures that self-assemble and things like this. From that we can try to infer from the sort of progression to through the trajectory of what might be happening, but always we're just trying to compare this to experimental data. In some cases, we do have intermediate structures. And again, it's somewhat hand-wavy, probably not a very satisfying answer, but we just try and check that it's reproducing what we know experimentally.